All right. Good morning again. I guess we're ready to start. So I hope everyone can hear me well. Good morning and welcome to our final microbiology webinar of Labconsult. So this is the last of 3 webinars we organized, and recordings of all 3 webinars will also be available afterwards to share, to rewatch. If you have other people that think are interested in having a look, these recordings will be available. If you didn't receive them in your mailbox yet, please send us an email and we will send you the link to the recording. Today our speaker is Dr. Enrico Tatti. He's the territory manager at Biolog, and he will talk to us about phenotypic identification and characterization of microorganisms. So the company Biolog was founded in the US in 1988, and the company is based on a patented technology to characterize and study the metabolism of microbial cells using colorimetric readouts. So it's a relatively simple readout, and this is actually the beauty of the technology, I think. But Enrico will tell you a bit more about this. The first and the most important application of the Biolog technology is that it is used for the identification of a large number of microorganisms. So these are bacteria, yeasts, or filamentous fungi. And the Biolog database consists of over 2,900 different species that can be identified using the technology. And this identification process can be done automatically as well using one of the platforms that Biolog has developed, called the OmniLog platform. And again, Enrico will tell you a bit more about this. So identification will be the main topic of Enrico's talk today, as this technology is often used in pharma companies to identify their environmental isolates themselves, so to do identifications in-house. Apart from the identification technology or the identification application, Biolog has also developed assays to study the metabolism of not only microbial cells, but also mammalian cells, and more recently, even also to have a look at the metabolic activity of specifically the mitochondria inside mammalian cells. So we will not cover all these different applications of Biolog in this webinar, but if you're interested, just send us an email and we'll get in touch with you to talk about these different applications, or we can set up a call with Enrico. There's different possibilities. So about our speaker today, Enrico, he is an experienced microbiologist. He obtained his PhD in microbiology from the University of Florence in Italy. And since then, he has worked as an environmental biologist in different companies in Canada and in Ireland, before he took up his role as a territory manager at Biolog. What he is today. As said, Enrico's talk will be mainly focused on the identification of microorganisms, but feel free to contact us if you're interested in other applications as well. Just want to mention that if you have any questions for Enrico or general questions during the presentation, feel free to type them in the chat and we will cover them after Enrico's presentation. We can discuss them with him. So please, I think that's it from my part. I think Enrico, whenever you're ready, you can start your presentation. Thanks you all, and enjoy the webinar.
Thanks. Thank you very much, Sam, for the introduction, and thank you everyone for attending today's webinar. As Sam said, yes, I'm Enrico Tatti. As you can imagine from my name, I'm presenting today from Italy, because that's my home country, and most precisely close to Florence. So bring you some greetings from Tuscany, which today is not really sunny, but generally it is. So As Sam said, today we will talk about phenotypic identification using Biolog technology, and I'll have also a few slides about how also microorganisms are characterized in depth with the same phenotypic approach that we use for ID. But before Sam already did a little bit of introduction on Biolog, I'll just extend it for a few more information for you, just to put it on the map. So Biolog is an American company. It's an American company. It was founded in 1985. So there's many years now that is in business. It's based in California, as you can see from these maps, on the sunny West Coast, and is located basically in the outskirts of San Francisco in Hayward. So a very nice place to go and to do our annual meeting. Although, you know, this year we were not able to go for the known COVID situation, but very nice places. And it's a business, as I said, that was born in 1985. It's an international business with active in more than 60 countries that we reach with the invaluable help of our distributor. And Labconsult is one of our most important. I'll say a few words in the next slides about that. It's a company that developed many different products. Around 70 that are manufactured in California, and also around these 70 there are 20 issued patents plus US patents plus other country that this technology has been patented. So the main target, the main technology and the main sector for Biolog is microbial identification, non-clinical microbial identification. But as Sam said earlier, we are also very present in life science research with our phenotype microarray technology, which span from microbes to human cells, and that I will show you just very briefly at the end of this presentation, just to give you a continuum from our first core business, which is a microbial ID, then further develop in phenotyping, extensive phenotyping and cell characterization. So as I said, the company is based in the US, but we do operate outside the US through the help of our distributor, and for the Benelux, for Belgium and Luxembourg and the Netherlands, Labconsult is our business partner since 2016. So I started in Biolog 2 years later, and I found myself in a very good group of people with Elizabeth Days and Linda Chabal, and then 1 year later, Sam De Decker joined us. And this is actually one of our most knowledgeable distributors, formed by people that have a high knowledge, PhD, and a lot of experience in the lab. So it's very easy for a company like Biolog to work in conjunction with Labconsult, because the knowledge that to explain and to expand the knowledge of our customer in Belgium is there through the people that work there. So it's a very good collaboration that we have since 2016 that we are very happy of. So after this brief introduction about Biolog and our partnership with our distributor Labconsult in Benelux, I'd like to outline now a little bit what is going to be in today's presentation. So I divided in three essential parts. The first part is an introduction to the concept of phenotype, and why the phenotype approach should be considered interesting. You know that nowadays, and since a few years, more cutting edge methods has been developed in analyzing cells, spanning from human cells to bacteria and fungi and yeast and so on. And everything that has to go with the molecular technique has a little bit more appeals due to the novelty of this technique and how fast they're developed. And these are very great techniques that we all know how fundamental they are in biological research and even further. But my goal today is to show you that even the concept of phenotype, although it sounds a little bit retro, it's still very important and very useful, and allow us to access a level of information that can be exploited for identification in a very clever and functional way, and further characterization of microbes, but not only microbes, also any type of cells. So after putting the concept of phenotype back in the map where I think it should belong, I will talk about how we can identify microbes using their phenotypes and their phenotypic features. And then the last part of the presentation will be very short on high-throughput Phenomic, which is an extension of our identifying technology. And then we'll have a Q&A session, which please to type the question as Sam said, and we'll be happy to answer not only today but also later if you have more. You can contact Labconsult or myself. My email will be visible at the end. So the concept of phenotype. So the concept of phenotype, I think we are kind of all familiar with what the phenotype is, and if we want to give a definition, the phenotype, the best definition are in our textbook, and they are still valid, although they need a little bit of update. Basically, the phenotype is the recipient for all the genetic info, and it's a Greek word that comes, from ancient Greek, that means display. So the phenotype is, has been taught, and for a long time, that is the manifestation of all the genetic information or the genetic makeup of an individual. So being a human or a plant or a bacteria whatsoever, it doesn't matter. That's what the phenotype is. Or if you want a more technical definition, is the observable physical or biochemical, which is important to keep in mind, not only physical, but also biochemical characteristic of an organism, determined by both the expression of its genetic makeup and also the environment of what this organism is in. So this is the classical definition of our phenotypes, and still it makes it important, but it places it a little bit on the marginal side of the flux of information, if you want. So if we look at the classical view, or also what we call nowadays the reductionist approach, and we'll see why it has this exception now, puts the phenotype at the very end of the flux of information, or at the very end of the dogma, central dogma of biology. So the information travels linearly, for the classical view, from DNA. There's a transcription and gets an RNA molecule that carries the information, and then it transcribes into a protein, and the protein then, combining with a substrate, gives a certain phenotype. And more often, the phenotype that we imagine from our knowledge from biology, the phenotype is, like the color of a flower, or the color of our eye, or if it's a bacterium, if it has flagellae, if you have fimbriae, anything that we can actually observe visually. But the truth is that this is a classical view that has been updated, and we need to consider that as in need of update. And it's also a very basic way to consider the traveling of information, which we know now that it doesn't travel so linearly and simply from DNA to the effect at the phenotype. Actually, the genotype and phenotype link is much more complex than that. And if you look at these conceptual maps present in this very interesting paper about phenomics by Barry Bochner, the CEO of our company, was published in Nature Review Genetics in 2003. In this conceptual map, you see two circles. The inner circle is actually, the red one is the genotype, and the yellow circle is the phenotype. And you can see there are different genes. So if the classical view would be true, and it's actually true, but it's not on the only situation, at one gene will correspond one distinct phenotype, but it's not always the case. There are many other different situations that can be present from when the information travel from the gene to the phenotypes. And these are a few examples, few notable examples that show how this link is much more complex than we initially thought when we look at the classical view of this central dogma of biology. So there are some times where a gene leads to actually two distinct phenotypes, or a gene leads to multiple phenotypes, or a gene is even silent. There's no effect at the phenotype, or as effect on distal effect on very distant and apparently unlinked phenotypes. So all that to say that this link is not linear, is not that simple, and it requires an update. And what it requires is a systemic view, or if you want, an holistic approach. It's too simple to look the information and think that the information travels from DNA and gets by RNA and protein and metabolites directly to the phenotype. Actually, we know now, since the newest research in the last 10 years, that DNA and RNA and proteins and metabolites also interacts with each other. And this interaction is very important and leads also and controls the cell phenome. So the situation is very much more complex than the linearity that we were taught initially, or we thought there was initially. So it's very complex. And this, although it's complex, it's very important to consider this complexity, because it's only looking at the complexity we can actually understand those that are called the emergent properties of a biological system, or those properties that comes when all the elements that carry information works in orchestration together. And they are not accessible if we study only DNA, if we study only RNA in separation, if we study only protein, we cannot access this information. So it's all much more complex than that. And if we study in isolation, we cannot reach and most of the time grasp all this complexity. So all of this to say, and this complexity is depicted in these slides. You know, we see that the phenotype is the effect of DNA, RNA, protein, and metabolites, but also the interaction between all these elements. And this is a new science that is called interactomics that study this. So it's all very, very complex. So the phenotype per se is also a complex thing to study. And that's why an angle as phenomics or the phenotypic approach may be very useful. Because allow to see all the downstream effect of all these interactions. All these downstream effects can be visible, for example, in metabolic pathways or cellular process or cellular networks. But most importantly, the phenotype, especially when it regards bacteria or unicellular organisms, it leads to the ecosystem function. And what is the ecosystem function? In fact, is what the microbes can do. What are the functions that a microbe can do? What are the metabolic features that a microbe has? So keeping that in mind, we can understand that looking at the phenotype is not as simple as just looking at the morphology of a cell, of a bacterial cell, but also what are the capabilities of these cells, the metabolic capability of these cells that are extremely complex, as we know. So all that to say that a phenotypic approach, together with the other approach, can be a very interesting angle to look things from. And obviously it's not that obsolete as we used to think. It's still very cutting edge and can give profound and very interesting angles that can be integrated with all the rest. So all this complexity come to the phenotype, all these function or ecosystem function or capabilities can be used for microbial identification. And I'm going to show you how Biolog got to this point before showing you how our technology works in details, just a little bit more conceptual explanation on how microbial identification can use all this phenotypic information to work with. So as I was saying, if we consider like a bacterial cells, all the biochemical pathways that bacterial cells can do are phenotypic traits, basically, belongs to the phenome of the microorganism. And it's a very complex metabolic circuitry of cell, which becomes very difficult to study from if you try to decompose it in the genomics, in the transcriptomics, and the proteomics. But you can embrace completely if you look actually at the ecosystem function or their functionality and the phenomes. So the first person, the first pioneer that thought that this could be, all this complexity could be analyzed by a phenotypic point of view was actually a scientist in the Netherlands called Dan Doren de Jong, which in 1926 proved that actually microorganisms could be distinguished based on their metabolic feature, and in particular by how they could utilize or not utilize different sets of carbon sources. So I'm going to show you now actually a publication from Doren de Jong in 1926 that he was able actually to define different species or Burkholderia based on their utilization or non-utilization or of several carbon source. Maybe the slides is a little bit too tiny for you to read it very clearly, but we have here different carbon source on this column here, and the other column are different species of Burkholderia that you can see by a plus or a minus, that if they were able to grow or not in a determined carbon source. So this was the base that Biolog developed 60 years later, based the differentiation of bacterial species, the identification also of bacterial species based on their phenotypes. Phenotypes that then is linked on all the ecosystem function, all the emerging properties that are coming from the integration of genomics, is genome, is proteome, and is transcriptome. So using all these phenotypic traits, going back to the biochemical pathways, what Biolog did actually was to make a discrete test, multiple discrete tests of different biochemical pathways that are present naturally in each microorganism that we could study. So for example, take the degradation of the utilization of a certain carbon source or a fatty acid or a carboxylic acid or an amino acid and test them simultaneously using a very simple tool, which is a 96 well plate. So you can imagine that if in a 96 well plate you add different ready compound, they will be able to stimulate and activate certain metabolic pathways if that microbes has this metabolic pathway. And focusing on that particular metabolic pathway or several different metabolic pathway, you can actually differentiate one microbe from another. So actually, the idea, the novelty idea after the work of Doren de Jong was to link cellular pathways to phenotype in the microbial world. So that's exactly what leads then to metabolic fingerprint. Metabolic fingerprinting that can be used and exploited to differentiate and identify the different microbes that they show different phenotypic feature. So again, you're seeing now one of the main elements of the Biolog technology, a 96 well plate, as I said before. And you can imagine that in this 96 well plate, in each of the plate, except for one negative control, there is a different compound that activates or could activate a different metabolic pathway that we see in the metabolic pathway map before. So all that you need to do is add your cell and also add a proxy, something that will flag to you if that metabolic pathway is active and how active it is. And that proxy is actually a redox dye that is able to produce a color if the bacteria are active in that particular well, meaning if that bacteria have that phenotypic feature of that metabolic pathway in their all their traits. So basically, that's how the rationale of the test work. You have this 96 well plate with different tests that activates and tests different metabolic pathway, the redox dye, and your cell. And that's all. That's the major component. And I'll show you now what, in a few slides, what the other most important component there is, this technology. So this is just to show you, this slides now is again a little bit conceptual, just to show you more in details how the redox dye works in concern with the technology. Here you can see a cell, and you can see the two main metabolic pathway in a cells, the glycolysis and the TCA cycle. So what happens if the cells finds in the well a compound like glutamine, it internalize it, and these activate the TCA cycle that then has a production or activation of the electron transport chain. And this leads to NADH production. This is a universal energy currency that we have in our cells, in bacterial cells, in yeast cell, fungi cell. That's all it works in all the cells, the animal cells from bacteria to human. And this NADH is able then to interact with the redox dye that I mentioned earlier. It's a tetrazoline redox dye. And reducing this redox dye, this dye passed from colorless, no color, to a purple color. In a way that is proportional to the activity of this reaction. So a lot of reaction on glutamine, a lot of NADH generated, more dye reduction. And this works either if we, even with other compounds, and maybe go through other main pathways in the cells. So glycolysis, the case is fructose. The end point results is still the activation of the electron transport chain and the production of NADH, which then can interact with the dye, reduce it, and there's a formation of color. So that's how it works. And again, this is a very universal mechanism. We have dyes that work with bacteria. We have dyes maybe that work with yeast. We dye with the fungi, and dye will work with human cells. But the rationale is all the same, because this actual reaction, these biochemical pathways are pretty much universal, as you know. So based on this concept of a plate, a dye, and the different testing compound, Biolog developed an identification technology. Identification technology nowadays is called Generation 3 identification technology, or Gen 3. So you can imagine that there's two earlier versions, but a technology that is essentially based on 96 well plate, and that is based on the metabolic fingerprint that is obtained by different microbes tested in the same conditions. So basically, you can imagine that we have this so-called Gen 3 identification plates that is arranged with several different tests. The Generation 3 comes with actually 71 carbon source assays and 23 chemical sensitivity tests. So here is it illustrated a little bit more in details. We have a section where there are simple sugar, with a section with amino acid, hexose acid, or carboxylic acid. And then there's the chemical sensitivity action with pH test, sodium chloride test, lactic acid, and antibiotic. Why this test has been chosen by Biolog? Well, basically because after many years of testing and R&D activity, these were found to be the most discriminating test that you can do. And you can see that this is this metabolic fingerprinting here with the purple color formatted here means that this function in this of sugars are active in Stenotrophomonas maltophilia, and these other white are not active. See, no color is formed here. There's also some faint color, which is very useful as well, because there's 3 levels, so use, borderline, and not in use. And this is actually the very unique metabolic fingerprinting of Stenotrophomonas maltophilia. So you can imagine that for different microbes, the metabolic fingerprinting will be extremely different. So you can see here an example of 4 different bacteria that are being tested on the same Biolog Gen 3 microplate. So we have Moraxella, we have Corynebacterium, we have Acinetobacter, and Streptococcus. And you can see that they have all different metabolic fingerprinting. And also this type of analysis can be not only providing information on the ID, on the unique ID, but also very important biological information. For example, you can see that Acinetobacter is not using a lot of simple sugar, but using more carboxylic acid, and it's very resistant to toxic chemicals, as you can see here on the metabolic, on the sensitivity test side of the plate. So this is we know about this, about Acinetobacter, but it add to the fact that actually this type of test does not tell you only a name of your strain, but also important biological information that can be of use. But again, all these slides is to show you how they can be different. And actually, you can imagine a Biolog together with this plate has prepared a database where your results that you obtain with your bacteria that maybe you don't know what it is will be run against, and the database will find a match, of course, if there is a match, and will tell you the ID. And that's how simply it works. So it requires a plate, and it requires software and a reader that I will cover in a moment. But before that, you can imagine that the number of combination of these tests are very, very high. Actually, it's a very high number because it's 2 to the 95 possible test combination that you do. And this means more resolving power. So a lot more tests means more resolving power, but also the ability to accurately identify many more species and to differentiate them at a very fine phylogenetic resolution level, even to the strain level. So that's why it can be very powerful. There's 95 tests, 1 negative control, as I said, that can lead to so many diverse molecular phenotypic fingerprinting that can differentiate all the species. And this is a little bit more details on the Biolog Gen 3 application. The Biolog Gen 3 application actually has in its database over 1,500 different species. And the database has been built and crafted to suit the needs of pharma quality control microbiology, veterinary microbiology, cosmetics, water analysis, food, agriculture, meaning that you can find in this over 1,500 species, you find microbes that are commonly found in this type of environment. So pharma, environmental monitoring, veterinary settings. So there are some pathogen for animals. There are some food pathogens, some plant pathogen, and also microorganisms that are found in water and cosmetics factories as well. And the system also is being used quite extensively, especially in university in the US for educational purpose and to analyze also complex microbial communities. But the core is the core of the database, what the database was actually covered. And we can share with you a full, complete list of what is in the database entails pharma, veterinary, cosmetic, water, food, and agriculture. So very, very important area. And another thing to consider that is a very interesting of this system are some fundamental key points. Apart from the extensive and phylogenetically broad coverage that is possible to obtain due to the high resolution power analysis, this Gen 3 plate doesn't require any pre-test. So you can use this single plate. The Gen 3 plate is good for gram-positive and gram-negative. So meaning that you don't have to do any gram staining before any pre-test of oxidase or catalase. The tends, they're fastidious to do, they're time-consuming, and sometimes they can lead also to an error. It's easy to make some mistake in this analysis. So one panel fits both gram-negative and gram-positives. and you don't have to do any other test when you are doing this bacterial identification. You don't have to do any other test after the end of the analysis or before the analysis. The nice thing is also, and I'll show you a little bit details later, is that there is a closed database of over 1,500 species, but the user can also make his own database, so it's a semi-open system without affecting the original Biolog database, and creating his own to suit his needs, to find and to follow some of the bacteria that may not be in the database. Although Biolog updates the database on quite a regular basis, we know that microbial diversity is so high that it's easy to find sometimes something that is not in the database at all. So you can add it. So a little bit more details on how this works from the preparation and experimental side point of view. So there are, as you can imagine, you need to work with bacteria that are isolated. There's no system that works with mixed culture, so you need to have an isolated microbe that you don't know what it is, and then all that you need is to grow them in a solid agar media, and then collect a very little amount of biomass. That's all that is needed. Most of the time, in most of the protocol, there are 3 different protocols depending if you're working with fast grower, medium grower, or slow grower. Most of the time, you need just 1 or 2 colonies. So very little biomass that is needed. And then the protocol can be done realistically in 5 minutes, for even less after you've done a few plates. And after you collected the biomass, you resuspend it homogeneously in an inoculation fluid. You reach a certain level of cells that are measured by turbidity, and then you transfer everything into with a multi-channel into the gentry plate. You incubate for a needed time, and then you read the plate and you compare it against what is in the database, in the Biolog database. And then the software will tell you the ID, will give you an ID, and will give you also a quality ID, a quality controllable ID that you can check and understand the quality of your identification, how good it is. So about reading the results, and these are the other very important components for the Biolog technology. So we have these plates, obviously, and that requires your microbe, the dye, and the inoculation fluid. But then you need a database, of course, and you need also a way to read your plate. So we do have an option for manual reads, which is called the manual system, but it's not used for QC microbiology. It's more in research and teaching. For controlled and regulated quality control microbiology, we do have 2 instruments that read the plates automatically. One is the microstation, which is called semi-automated, because it's like a 96 plate reader, where you add 1 plate per time at defined time, and you read, and then the microstation send the results through the software, and then it's compared to the database. Or you have a fully automated solution, which is called the Omnilog, where which is an incubator reader that keeps the temperature. You prepare your plate. The plates are placed inside the Omnilog from time 0, and the Omnilog automatically reads the plate every 15 minutes and sends the results to the computer for to run it against the database and see if there is a match. And this is done until there is a match between your profile, your metabolic fingerprint, and what is in the database. So in this way, you don't have to manually measure into the microstation reader, but you can set up the experiment and walk away. You can set it up in the evening and the morning, finding the results. You can set it up in the Friday and find the results on Monday. And the nice thing is that you can you can put inside the Omnilog up to 50 different plates. So it's a very high throughput. While in the microstation, you can analyze, you can read multiple plates at one per time and moving them from the incubator to the microstation. So it's a less automated system. But these are the best option for QC microbiology because they have the option of also validation, and they have also software that is compliant with 24 part 11, CFR part 11, as I will say in a little bit more details in a little bit. The results also, I want to, before I highlight other feature of our ID technology that regards not only bacteria, but also fungi, yeast, and anaerobes, I want to, for a second, stop on the outcome, the results that comes from Biolog technology. So Biolog technology doesn't give you only a name, but it gives you the capability of QC and understand how good your results is. So you can see that this is how the results normally look like. There's a profile that is obtained, metabolic fingerprint, if you want to call it, is obtained analyzing one unknown strains, and you can see that it gives a Staphylococcus capitis species, and then the software gives some measurements, some precise measurement, or how good this matching is, how much it overlaps with what is in the database. It gives few options that could be possible for the ID. The first one, of course, is the best fit, and others, and also allows you to understand where your result doesn't align with the database. So it's very easy to understand if you're working with a bacterial species that maybe is stressed, or maybe it's very easy to even to understand if you are working with a mixed sample. If you have, for example, two different bacterial species that are even difficult to see. So interpreting the result doesn't stop on giving you a name, but also gives you the possibility to quality control your results. And this is very important, I think, in the QC microbiological settings. So along the same line, after Biolog developed the Gen 3 plate, after a few years, Biolog also realized that the same technology could have been applied to identify filamentous fungi. And so it developed the so-called FF microplate. FF stands for filamentous fungi. You can see it works, it seems pretty much as the Gen 3 plates. The only thing is the different color of dye. It's still a tetrazoline-based dye, but we call it salmon red dye, as a slightly different chemistry to work well with the biology of filamentous fungi. But essentially, the rest of the plate is it's very much the same as a different substrate in each well, a negative control. You add your filamentous fungi in, and then you incubate over time. And if there is a metabolic activity, you will see it, and you will obtain then a metabolic fingerprint that you can run against the database. In the database, there are over 700 different filamentous fungi, 710 to be precise, and which makes Biolog the largest database, the biggest database in the present for the moment in the market at the current time for filamentous fungi. So the way the plate works is basically the same as the bacterial one. Obviously, fungi, who works with fungi knows that it's not as simple as working with bacteria. They have probably a better smell than bacteria, but they are a little bit more difficult because you have sporulating fungi, non-sporulated fungi, but obviously Biolog has accommodated this in his protocol, so you can work with both sporulating and non-sporulating fungi with just a little bit different approach to the colony morphology. But the rest is the same. You get your biomass, these are conidial spore or mycelia that are scrapped from the agar surface, and you resuspend it, you make an homogeneous suspension in the inoculation fluid, and then you inoculate the plate, incubate over time, and then get the profile that you run against the database. But whoever worked with fungi, as I was saying earlier, knows very well that fungi identification is not as straightforward as bacterial one. It's actually much more complex. Actually, anybody that works with fungal identification more often do not rely only on one single identification method, but try to use different identification methods to confirm and reinforce their ID, especially when this ID is crucial in for example, as in QC microbiology, in food or in pharma, or even agriculture, as you can imagine. So all that to say that since fungal taxonomy is very complicated, there is a large physiological and morphological diversity. So people that want to identify fungi, they normally, as I said, use maybe a genetic approach, and so they sequence their ITS, which is not very simple to do, as you may know it, and then maybe they do morphology studies. So they use two different approaches to reinforce the results and make sure that it is. So the same, you will be able to do the same thing with Biolog, meaning that you will have together with the phenotypic test, you can also do a morphology test. That's because Biolog FF database comes with the microscopic and microscopic images of all the 710 fungal profile that are in the database. So that means that after you get an ID, for example, the system gave you an ID for Aspergillus puniceus, for example, you have the chance to access the FF image library that is in the database. It's easy to access with a few clicks, and see what are the morphology of this fungus. So in this way, you can go back to the original plate where you had the fungus, take one sample, put it under the microscope, and see if there is an alignment with what you see in the image library and what you see under the microscope. And that's a very powerful way to confirm and reinforce your ID and making sure that you didn't make any mistake. And together with the microscopic images, there are macroscopic images of the plates that shows the morphology of the fungus on different agar media, for example, multi-extract agar on the CYA agar after 7 days. And putting together this information from macro, micro, and the phenotypic approach, which is a biochemical, in this case, approach, you can get to a point where you can tie your ID to almost 100% certainty. So this is very, very important and very unique feature that Biolog gives together with the FF database. So Gen3 and FF database are probably the most known product from Biolog. As I said, Gen3 is 1,568 different bacteria. FF microplate 710 fungal taxa. Then we have other plates and other databases that I didn't explore here in particular because they work pretty much in the same way, but they focus particularly on anaerobes. So we have a database specific for anaerobes with 361 anaerobic taxa. And also we have a specific database for yeast with 275 different taxa. So again, this is the system, can be run all in the same machine. For example, everything on the microstation, and you can add only the Gen3 for bacteria, or you can have only fungi, depends on what you're interested for, or you can have them all in the same system. So it really suits your needs. And as I said, as the possibility of adding also what is not in the database, adding it to the system through a separate program, which is called Retrospect. But also, as I mentioned before, and I think this is very important for all the regulated customers that we may have in the Benelux, our software and system are all 21 CFR Part 11 compliant. That's because the software, they have all the required security feature. You may be, familiar with this, the password expiration, the user privileges, the log on notification failed and attempted login, login archive, and also all the electronic recording integrity, like limiting access, documentation of changes, origin record integrity, and all that can be used for the audit trail. So this is very important, and we, as Biolog, we always put a lot of effort in making sure that this is maintained and kept up to date, because we know that our most important customer, the regulated customer, are require this in particular. And also, I want to mention briefly, we have very established system validation options that include validation manuals for the different database that we have. We also work in conjunction with other companies that have, for example, type strains, and these are microbiologics, validation supplicants. And we also provide, together with our distributor on-site validation services for all the different options that you have seen earlier. So all that to say that the Biolog technology is a full package, doesn't come alone with the technology, which is obviously very nice, but also fulfill the needs for a regulated customer, that we know how this can be important, and we continuously work and support our customer in these key aspects. So all that to sum up, I got to the end, to the ID part. Biolog, I think, is much more than an ID system. It allows you to identify, if you put all the database in the same computer, over almost 3,000 different species. That includes the anaerobes, the yeast, the bacteria, and the filamentous fungi. And the phenotypic system, the analysis per se, gives you an ID, but also provides biological relevant information that you can use to go to a resolution that is at the strain level most of the time. This is really important. So it doesn't give you only a name, but it gives you also biological information that are important for strain level resolution, but also to QC your own results. So you can understand if your results is good with Biolog, and how good it is and how trustable it is. It's not just a name. And as I said, the last thing which is important for some of our customers is the database is a closed database that you cannot change or play with, but you can create your own database that you can run together or separately for all those bacteria, yeast, fungi, and anaerobes that are not included in the original database. And you can then use our software that comes together with the system, which is called Retrospect, to create this database and do tracking and trending on the ID that you do and find in your own setting. So a very complete package is together with all the validation and the 24/1 Part 11 compliance. I think it's can be particularly interesting. So this is pretty much all for the ID. Before I finish, I just want to show you how then Biolog developed further this technology. It may be of interest of you or for some of your colleagues, and how we transition from phenotypic identification to extensive high throughput phenomic characterization. And it's only a few slides. The transition was actually very smooth, because already we were characterizing in a way, in the same time that we were identifying, as I told you earlier, we were also characterizing, meaning that we were finding out biological feature of our bacteria, yeast, and fungi. So all that we did was to expand, to get into the high throughput phenotypic characterization, was to expand the array of microplates, meaning that beyond plates that test carbon sources, we have plates that test nitrogen sources, phosphorus and sulfur, or pH and different osmotic condition, antimicrobials for sensitivity, antifungals, heavy metals, mutagens, and so on. So we expanded all the possible tests from 95 to almost 2000. And this is high throughput phenomics, is the technology that I mentioned initially, which is called phenotype microarray. which works in the same way as the ID. You take your cell, you resuspend in inoculation fluid, you add the dye, you add it to the plate, and you incubate over time. But this time, instead of getting an ID, you get a very high resolution phenotypic characterization. And to do that, it gets really powerful when you use the instrument called the Omnilo that I showed you earlier for ID, that has these high throughput capabilities and keeps the temperature. You can put up to 50 plates. The Omnilo measures every 15 minutes automatically and reads automatically each plate. And then for each plate is able to give you the kinetic color generation, which is a kinetic measurement of the color production, or the respiration, if you want to call it that way, and provide you a high resolution analysis that you can imagine with the kinetic level can be very, very powerful, because there's not only yes or no qualitative analysis, but also quantitative and kinetic. And from the kinetic, you can export a lot from a single curve. You can export so much information, and you can imagine that this information multiplied by 95 and for the number of plates that I showed you earlier. So very, very powerful. And just to conclude, this technology has been used extensively now. It was developed in the early 2000s. It was developed for comparing initially two different cell lines or two different conditions, and compare them with the software and see immediately the metabolic differences. Just to conclude, these are the main application for this technology: testing cell lines, for example, mutants and wild type, and see the effect of a mutation on the phenotypic level. Testing what is the mode of action of a compound. If you treat a bacteria with a certain antibiotic, what is the effect of that antibiotic at the metabolic level? Or also characterize more in depth bacteria or fungi or yeast for biotechnological application, optimizing them growing condition, finding the most the preferred carbon source, nitrogen source, device selective media, and so on. So a very comprehensive technology that is done with the very same machine. The Omnilo can be used for identification and for characterization, just switching from one software to another. So it's a very universal technology that can be used in multiple ways. So before I finish today, I want to thank you, of course, for your time, but I also want to thank you greatly, Labconsult, for organizing this webinar series. I think they were really interesting, for allowing me to present you and illustrate you how the Biolog technology work. As I said, we have a very dedicated and knowledgeable team in Labconsult, which is really easy for us in Biolog to work with, and we are really happy to work with it, particularly Linda and Sam, that they cover different area of the Benelux. They have a very high knowledge of our system, and they can explain it to you very well. So just feel free to reach out to them for any further explanation that you may need on this system. But also I'm available as well. That's my email. If you want to reach out, even after this webinar, I'll be very happy to send you material, presentations, or answer any of your questions. So thank you very much again for your time today, and I'm happy to take any question now.
Thank you, Enrico. It was all very, very clear, very interesting talk. Thank you.
Thank you.
So any questions by the audience? During the presentation, we didn't get any questions in yet in the chat. I'm not sure if anyone else at this stage has any questions for Enrico about the Biolog technology in general, or specific applications they have in mind. If not, I think the bottom message or the basic message can be that if you have any interest or if you have any questions, or you're wondering how you can apply the Biolog technology for your assays, for your research, you can just contact us at Labconsult or contact Enrico directly, and we'll happily talk you through the different possibilities. I think, correct me if I'm wrong, Enrico, but I think a strong point of us and of Biolog as well is that we try to think with the customers about their specific projects, because often specific projects have specific questions that they want to see answered, and there's a lot of different possibilities with the Biolog technology, as you can see through in this presentation. So we're very happy to discuss your specific project and see how we can apply Biolog in the best way to get the answers that you are looking for.
Yes, I agree.
So at this point, I don't see any questions coming in yet. So I guess either it's very clear, everything is very clear, or the people are just a bit shy, and then hopefully they can send us an email or contact us later to discuss anything that they would like to discuss.
Of course, so, anytime.
Thank you, Enrico. Thanks again for giving us this very interesting talk. People that are interested, again, please just contact us. And then I just have to thank all of you for attending these different webinars. I've seen many of you have attended all the 3 webinars, so the 3 subsequent weeks. So thanks a lot for that. I hope it was a bit interesting. And let's hope that next year the conditions allow us to do a physical meeting again, like we did previous years. So I guess that's it from our side. Thanks again. And again, we will be in touch via email with the link to the recording of this webinar and also the previous webinars. If there's anything else, you know where to find us. And I would just say thank you and hope to see you all soon.
Thank you,
Enrico.
Thank you, Sam.
Bye. Talk to you all.
Bye
soon.
now.
Bye.
Bye. Have a nice day, everyone. Bye.